This conference will now be recorded.
Hello everyone. Today's topics will be vitamins and after that hormones and then nucleic acid. So these three are the main biomolecules that we'll discuss today and also related problems. So what is vitamin? They are small organic molecules, cannot be synthesized by our body, but essential for certain function. So as we cannot synthesize them inside the body, obviously we have to take it externally, and that is through diet. And what is the requirement amount? Is it required in high amount or low amount? Its presence which is required, that is very low amount. Okay, but remember, even if it is very low, but if this low amount is not maintained, then there will be deficiency problem. That is, if it is lesser than the amount which is required, though the amount is very small, but if it is still lesser than that inside the body, then there will be some problem. And also, and there is another problem, if it is present in excess, so there should be some optimum range which should be maintained, that is amount of vitamins. If it is lesser than that, then there will be deficiency problem, and that will lead to some disease. And if it is more than that, then also there will be some problem, which is because of excess amount. Now vitamins, what are the different types? They are basically designated by different alphabets like A, B, C, D, etc. Okay, so here we use the alphabets A, B, C, D, etc. There are other alphabets also, but these four are the main vitamins. Now there may be some subgroups. For example, if you consider B, under B we can have 1, 2, 6, 12, etc. Okay. Now each vitamin, whether it is A, B, C, D, like this, there is specific function in the living system, and mostly they act as coenzyme. Now what is enzyme? That we have already seen in the last class. What is enzyme? That we have seen in the last class, but now we are saying that they behave as coenzyme. Now what is coenzyme? So coenzyme is basically defined as an organic molecule that binds to the active site of the certain enzyme as an assistant. Okay, so as if coenzyme is assisting in the role of enzyme. Okay, so enzyme is basically biological catalyst, and coenzyme, when it is associated with the enzyme, they are basically assisting in their whatever role the enzyme is playing. So as coenzyme, what they do, they are responsible for growth and development, then repair and healing wounds, maintaining healthy bones and tissues, for the proper functioning of immune system and other biological functions. So these are some of the biological functions that you are seeing here, and they are not served as energy sources like carbohydrate, lipids, etc. So carbohydrate, lipids, they are basically source of energy. Okay, in presence of oxygen, these carbohydrate, they are broken and energy is liberated. So that is why they are energy source. But vitamins, they are not energy sources. That is the important difference with carbohydrate molecule. Classification of vitamins. We can do the classification based on solubility. Okay, now some vitamins are soluble in fat, some vitamins are soluble in water. So two categories are there. So here you can see, when it is water soluble, under this category we have vitamin B and C, and for fat we have A, D, E and K. So when it is fat soluble vitamins, these vitamins obviously will be absorbed best when taken with fatty food, because they are soluble in vitamins. And another reason, another effect is that they are stored in fatty tissues and livers. Okay, so these vitamins, they are not soluble in water, and that is why they are called fat soluble vitamins. And here you can see this example. Now as it is not water soluble, so we don't have to take this vitamin in a regular basis, because when it is water soluble, there is a chance that it will be excreted through urine, but that is not the problem here. And what about water soluble vitamins? Vitamin B, and under B you can see all these varieties are mentioned, and C, they are readily soluble in water. Now that is the reason if there is any excess vitamin, they will be excreted through urine, and it is not possible to store in the body. Only one exception is there, which is vitamin B12. So that is the reason these two vitamins, that is B and C except B12, it should be supplied regularly to our body. We should take it on a regular basis because they are water soluble and there is a chance that it will be removed through urine. Okay, so that is why it should be taken regularly. Here we have all the 4 columns, and in each column the vitamin name is mentioned. Then what are the sources from which we can get this vitamin, what is their role, functions, and deficiency disease. That is, if it is not present enough amount, what will be the disease, that is also mentioned. So there is nothing much to discuss. You just have to have a look in these 2 charts, and here you can see under each vitamin name there is another name mentioned which is within bracket. Okay, now these names are basically chemical names of vitamins. So see, vitamin A, retinol, calciferol, tocopherol, like this, there are different names present. So why it is some of, at least some important vitamins like retinol, calciferol, tocopherol, why it is important to remember, because sometimes in problem, instead of mentioning the name like with alphabet, I mean A, B, C, D, if it is mentioned like retinol or tocopherol, then we have to understand which vitamin it is talking about. Okay, so at least this that I am marking, try to remember, because under B there are so many different types, so at least try to remember these 4 to 5 vitamins, their chemical names. Okay, so that in problem, if these names are used, you will not have any problem. So after vitamin, the next important biomolecule is hormone. So what are hormones? They are chemicals that essentially function as messenger of the body. So their role is basically messenger to that is spread the message from one part of the body to other part. Okay, now these chemicals directly secreted into the bloodstream by special glands. So there are some special glands that are responsible for the secretion of hormones, not just any gland, and these glands which are responsible for secretion of hormones, they are known as endocrine gland. And there are different types of endocrine glands. They are distributed all over the body. Now blood carries this hormone to different organs and tissues so that they can play their role. Okay, so these messengers, that is hormones, they control different types of not only physiological but also psychological. Both these functions they control. Now what are the functioning of hormones? Some that is main important functions of hormones are mentioned here, and you can see here both psychological as well as physiological functions, both are present. If there is hormonal imbalance, that is if it is excess than the required amount of it, if it is lesser than the required amount, then there will be some imbalance. Now because of this imbalance, there will be excess or deficiency, and that will lead to actually some disease. So that is the reason a proper balance should be maintained if we want to have a healthy body. So just like vitamins, we have seen that a particular range should be mentioned. Sorry, some particular range should be maintained. Here also same thing. Proper balance must be there. It should not be more, should not be less. Now classification of hormones we can do in different way. So the first classification that you are going to see, it is based on the distance over which they act. That means they are produced at some position and then they are acting in some other position. So these 2 points, what is the gap between these 2 points, that we have to based on that we can have some different types of hormones. So the first type is autocrine. Now in this case, the cell targets itself. So here you can see a single cell is present. So the point where the hormone is generated, it is acting in the same cell. So it is autocrine, auto. That means within same cell it is playing its role. Now the next one is signaling across gap junctions. Now gap junctions, it is a particular biological term which is used. So between two cells, if there are some intercellular channel, that means as you can see, these are some channels through which the cytoplasm from of one cell, it can be connected with the cytoplasm of the another cell. So the left hand side cell, that is the green one, is the signaling cell where the hormone is actually produced, and the right one, which is the pink colored cell, that is actually the target cell. But these two cells, how they are connected? By gap junction, that means some intercellular channels or tunnels. So these types is another type, second type. Now the third one is paracrine. A cell targets a nearby cell. So here there is some gap between the signaling cell and the target cell. So the hormone is produced here and it is playing its role in the target cell, and there is some distance between these two cells. And the last type is endocrine. A cell targets a distant cell through the bloodstream. So the previous one, that is paracrine, these two are as if neighbor to each other, but endocrine, it is far from it. So now the hormone, it is produced in the signaling cell and target cell, and it is moving this large distance obviously through bloodstream. So these are the four possibilities, and that is based on distance over which they act. The second classification is based on chemical composition, that is how they are formed, what is their actual structure. Depending on that, there may be here you can see three broad categories: steroid, amines, and peptides. And under steroid we have total 4 hormones. Not that is the main hormones are here given as example. Under amine, thyroxine, adrenaline. Under peptide, insulin. When it is steroid, they are actually fat soluble molecules that are made from cholesterol. So cholesterol is a very large organic molecule. Now among these are the 3 major sex hormones. So these 3, and steroids, they pass into the cell's nucleus, bind to specific receptor. That means it is working in the nucleus of the cell, not cytoplasm, and it is binding to the specific receptor and genes, and ultimately trigger the cell to make protein. So this is the role of this steroid hormone. But remember, basic structure, it is made from cholesterol. The second one is amine hormone. So as the name suggests, they are actually amino acid derivative. So their structure, they are amino acid derivative. And some examples such as epinephrine, which is water soluble molecule derived from amino acid. These hormones are stored in endocrine cell until it is needed, and then they can act by how they act by binding to the protein receptor on the outside surface of the cell. So see, in case of steroid hormone, what we have seen, it is interacting with the cell's nucleus, but now it is outside the surface of the cell, it is binding with the protein receptor. And the last one, peptide. Insulin, very important example under this category, which is basically that is the first example that comes in our mind. It is that is for peptide hormones, it is insulin. So insulin, growth hormone, then prolactin, which is another example, and other water soluble polypeptide hormones. So their structure is basically polypeptide. Amino hormone is amino acid derivative, but now it is polypeptide. There will be different peptide linkage, consists of long chains of amino acid. So it has to be long chain because it is polypeptide, and there may be several to 200 amino acid long. So see how long it is. They are also stored in endocrine cell, just as you have seen in the previous case, and until it is needed to regulate the processes such as metabolism, lactation, growth. Okay, so these are the 3 types, and that is based on the chemical composition of the hormones. Now the next biomolecule, it will be the last biomolecule, that is known as nucleic acids. This is very important because the inherent characteristic of each and every species, they are actually transmitted from one generation to another generation, and this is actually related to nucleic acid. Okay, so it has been observed that the particles in the nucleus of one of the cell, they are responsible for the transmission of this characteristic. We know that in the nucleus of the cell there are different types of particles present. So one of these particles is basically responsible for the transmission of this type of characteristic, and this particle is basically chromosome, and chromosome is made up of two things. One is protein and the second one is nucleic acid, and this is our topic of discussion. Okay, so remember the first point, inherent characteristic, it is transmitting from one generation to another, that is based on chromosome, and chromosome is made of protein and nucleic acid, and nucleic acid we will now discuss. There will be two types of nucleic acid. One is deoxyribonucleic acid, and that is why the name in short is DNA. DNA. Another one is ribonuclease, that means RNA. Okay. Now, nucleic acids are basically one type of polymeric molecule. So obviously it will be long chain. And if you when we say the word polymer, obviously there will be some monomer, and each of these monomer, they are connected to each other, and that is why we are getting the polymer. So if nucleic acid is the polymer, so the monomer is nucleotide. So several nucleotides, they are connected to each other, they are the repeating unit, and you are getting nucleic acid. That means another name can be polynucleotide because it is the monomer. So that is the reason nucleic acids can also be called as polynucleotide. Now this nucleic acid, if we consider their picture, RNA and DNA, though these two will be discussed in detail separately. So here you can see the picture of RNA, which is ribonucleic acid, and DNA, which is deoxyribonucleic acid. One important difference you can understand between these two, that in RNA there is only one chain, like right, but in DNA there are two chains. That is the important difference. But now we will go into the details of the structure of nucleotide. Okay, so nucleotide is the monomer, and nucleic acid, that is the polymer. So nucleic acid, as they are biopolymers of nucleotide, so when we do controlled hydrolysis of these two nucleic acid, we get mainly three components. That means these three components that are present in the monomer unit of nucleotide. Okay, so what are these three components? The first is nitrogenous base. Nitrogenous base means there will be a heterocyclic molecule which will contain nitrogen atoms. Fine. So it is a ring structure with nitrogen atoms. Second component is pentose sugar. Now pentose sugar means, remember when we have discussed sugar, and there is cyclic ring structure of sugar possible, and two types of structure mainly we have seen. One is 6 member, another one is 5 member. So when it is pyranose, it is basically 6 member. When it is 5 member, then mainly we have called it pentose. Okay, because total 5 atoms will be there. Now pentose sugar, when it is RNA molecule, the name is ribose. The pentose sugar is deoxyribose when it is DNA. But in general, you will call it pentose sugar. And the third component is phosphate group, which is inorganic part. Okay, so one by one we will see what are these three components in detail, because these three components together will form the nucleotide, and then several nucleotides connected to each other, you will get finally the nucleic acid. Nitrogenous base, the first component. There are they are basically nitrogen containing organic compounds, and you can call them heterocycle. Remember pyridine, it is just like benzene, but one carbon is replaced by nitrogen. So here also you will see something similar to that pyridine, though it is more complex. So there are mainly two types of nitrogenous bases. One is pyrimidine, another one is purine. So these two are the structures of purine and pyrimidine. Pyrimidine is just like pyridine, but here one nitrogen is extra, and the position of these two nitrogen, their positions are 1, 3 position. Then purine, here we have 6-member ring with 2 nitrogen fused with a 5-member ring again with 2 nitrogen. So this is purine, this is pyrimidine. Now under purine category, we have some bases. Under pyrimidine category, we have some bases. That means now we will have some derivative. Derivative means there will be some extra group or slight change in the structure. So when it is pyrimidine, under that you have first let me finish purine. Under purine you have two major bases. One is known as adenine in short capital A and guanine in short capital G. Now both these purine bases both are present in DNA and RNA. Now when it is pyrimidine, three different types of pyrimidines are there. One is cytosine, another one is thymine, another one is uracil, and here it is A and G. So A and G both are present in DNA, but when DNA and RNA. But in case of pyrimidine bases, among C, T, and U, only cytosine is common for both DNA and RNA. So cytosine is present DNA as well as RNA. Okay, but when it is thymine, then it is present only in DNA. And uracil, that is present only in RNA. Okay, remember this is very important information that T is present in DNA, not in RNA, and U vice versa. Now if we see the structure, so purine, we have this A, this is G. So see the basic structure, basic moiety is same. It is 6-member ring fused with 5-member ring. In both the ring you have 2 nitrogen. But difference is here we have this amine group 1 degree, and here also we have amine as well as we have carbon. Okay, and under pyrimidine category we have thymine in short T, we have cytosine, we have uracil. Here also if you look at the basic structure, though it is not aromaticity is lost, but if you look at the basic structure, all of these three are 6-membered with 2 nitrogen, right, at 1 and 3 position. Though in some cases it is NH, in some cases it is double bond N, but basic moiety is same. And in thymine we have these two carbonyl, then methyl, then cytosine we have amine and carbonyl. Here we have 2 carbon. So these are the difference in group. So that is why these three are different bases, though basic structure they are derived from pyrimidine. Okay. Now detailed structure of these is not required, but what is required to remember is that under purine, what are the two types of bases? Adenine and guanine, and under pyrimidine, thymine, cytosine, uracil. This much information you have to remember. And another information which is this one, that is DNA is present only in thymine, because question regarding this is very common, and uracil is present only in RNA, not DNA. So this is all about nitrogen base. Why we are calling it base? Because nitrogen is present. So just remember the structure of pyridine. We know pyridine is very, though it is not very strong base, but in organic chemistry, this is used as base. Okay. So just like that, they can also, that is purine, pyrimidine, because of presence of nitrogen, there will be lone pair and there will be some basic property. Though it is not strong base, but as there is basic property, so that is why we are calling it nitrogen base. The second one is pentose sugar. Nucleic acid had 2 types of pentose, and depending on that it will be DNA or RNA. So when it is DNA, then you will have this type of pentose sugar. If you look at the numbering, this is 1, 2, why it is prime, that you will understand when you will see the full structure. 1, 2, 3, 4, 5. Okay, now at 2 position there is no OH, but you have H. That means at 2 position it is deoxy. Deoxy means oxygen not present. That is why we are calling it deoxy. Just remember, when we say hydrogenation, it is addition of H to double bond. When we say dehydrogenation, that means it is removal of H. So when we are saying D in front of any other term, that means it is not present. So 2' deoxy, that means it is not present, which is not present, and 2-deoxy D-ribose. This is the full name. And for another one, that is deoxyribonucleotide unit of DNA. Deoxyribonucleotide means, remember, nucleotide is the monomer unit. So the monomer unit, when it is containing this type of pentose sugar, you can call them deoxyribonucleotide. And the nucleotide which is containing ribose as pentose sugar, that will be called ribonucleotide unit of RNA, and this is known as D-ribose. And D, remember D and capital L that we use for sugar molecules. So in this case there is OH group present, but here no OH group, and that is the difference between these two. Except these two, okay, one, that is 2' position, there is no other difference. And the last component, phosphate group. So phosphate group is basically this one, as you were seeing, P double bond O, and there are 3 single bonded oxygen, but these 3 side they are connected with some other moiety. But this is the basic structure of phosphate group. So phosphate, we are saying phosphate because it is not complete structure, it is attached to some other. But suppose if I am writing here OH, then it will be phosphoric acid. But it is not phosphoric acid, it is O then connected to some other group. Phosphoric acid forms phosphodiester bond between nucleotide, that you will see in next slide. So these oxygens, they are actually the connecting point between different monomer unit. Based on the number of phosphate group present in the nucleotide, they can be classified as mononucleotide, dinucleotide, or trinucleotide. Okay, so see, now if I combine all these components together, it will be something like this. So first attach nitrogenous base, which is here in green color. Now nitrogenous base, though it is true that it may be 6-membered for pyrimidine and it is fused ring of 6 and 5 for purine, but for simplicity it is simply given as 6-membered ring in this particular picture. So this is nitrogenous base which is attached to the pentose sugar, which is in blue color, and it is 5-membered. Now these two components together, that is known as nucleoside. So in this slide, the topics is nucleoside and nucleotide. So what is the difference? When you combine sugar and base, that is purine or pyrimidine base, then together we will call it nucleoside. But now when you also attach the third component, which is the phosphate group, now the name will be nucleotide. So nucleoside, it is already combining these two. Now you are adding the third component and the name is nucleotide. So this is the difference between nucleoside and nucleotide, that is presence of phosphate, whether it is present or not. And once you get the nucleotide, now n number of nucleotide together will make the polynucleotide, which is nothing but nucleic acid. That is the ultimate biomolecule that we are actually discussing. So now you can see 3 colors are used to denote individual nucleotide. So the first part in green color, that is a complete nucleotide. Now the second part in orange color, that is a second complete nucleotide. Then the third one, it is which is in blue color, that is another complete nucleotide. So here, as it is mentioned in the previous slide, that how many phosphates are present, or you can say how many monomer units are present, you can call it mononucleotide, dinucleotide, or trinucleotide. So in this particular picture, obviously it will be trinucleotide because you have total 3 monomer unit and also 3 phosphate. Now, if we see the connection point between these 3 parts, so this is one part. We change the color. So this is one part. Then another part. Okay, now see how it is connected. At 3' position, the oxygen that is present, and at 5' position, remember what is present? CH2H. Now this CH2O, as if initially there is as if OH present, OH present. And if you consider the phosphate, first you can write in this way, that is in phosphoric acid form. Now as if these H and OH are removed, and now you are getting this type of linkage, that is carbon oxygen, because this OH is attached to carbon, and when you are removing this OH and H, that means ultimately water is removed. Now you are getting POC linkage, which is at 3' position. So see here, this position number is important. So the OH group which is present at 3' and the OH which is present in the 5' carbon, these two are the basically connecting point. Similarly, the second molecule, that is the orange molecule, it is attached to the green one through 5'. But the orange nucleotide, that is also having 3' OH. Now this 3' OH is again used for connecting the third nucleotide, which is through this 5'. Similarly, this 3' also can also be used for the fourth nucleotide. Okay, so how these three are connected to each other? It is through the phosphate linkage, or you can also call it phosphoester linkage. Okay, so here phosphorylation, this process you can call it phosphorylation, that is individually first you can write the three nucleotides separately. But suppose when you are doing this removal of water molecule, now the process you can call it phosphorylation. So after phosphorylation, this individual monomer unit, I mean to say individual nucleotide, they will be now connected to each other. So phosphorylation occurs generally in the 5' OH group of the sugar. Okay, so 5' OH group of sugar, these three. So this is sugar, because 5' carbon is part of sugar. And nucleotides are linked in DNA and RNA by phosphodiester bond. Diester, why? Because two POC linkages are formed. So phosphodiester bond is formed between 5' OH group of one nucleotide and the 3' OH group of the another nucleotide that we have already seen. Okay. Now detailed structure of DNA, though detailed structure of RNA is not included in syllabus, we will have very initial primary idea, but structure of DNA we will have to see. So information regarding the sequence of nucleotide, that is which nucleotide is coming after which nucleotide. So here if you are thinking that what is the difference between these three monomer units, there is no difference. So there is no difference because here we have taken the same base, but it is not necessary. Maybe here you have adenine, then the next one is not adenine, it is something different. Suppose it is guanine and then it is thymine, it may be. So when different bases, because there are total 5 possibilities, and when it is DNA there are total 4 possibilities. So you can take any sequence AGT or ATG or GTA. So depending on this, what type of sequence you have taken, because only this base which is making the difference in each monomer unit, because otherwise there is no difference. Only difference is in this basic part. Here we have taken same base, but that is not necessary. So this sequence of base, whether it is AGT or GTA like this sequence is basically the primary structure of nucleic acid. Okay, but nucleic acid can also have secondary structure. Okay, now the secondary structure is double helix, which is formed by two polynucleotide chains. So the chain that you have seen in the previous slide, though there are only 3, but in this way if you continue the chain, it will be that is several nucleotide together, it will be one polynucleotide. Now take two polynucleotide, and if they are forming double helix to each other by twisting the structure, then ultimately you are getting the secondary structure. And how you are getting this type of twisted structure? It is because of the hydrogen bond that is formed between two helices. Okay, and the bases are bundled within the helix. That means bases will be remaining in the that is inside the helix. So if you look at the right hand side structure, it is a non-twisted form. So this is one nucleotide, polynucleotide. This is second polynucleotide. One polynucleotide. This is 5' position. That is the starting point. Ending point is 3'. And the other polynucleotide, it will be reverse. It will be 3' and then it is 5'. Okay. And this is actually not the real structure. It is just given so that you can understand it clearly. And you can see here we have different bases connected to each other, and this connection is basically through hydrogen bond. So guanine associated with cytosine GC, then TA, then AT, then CG. So you will always get these two types of pair. One is GC pair or CG, another one is AT or TA, because these are the four bases that are present. Okay, so these two types of pairing of base you will always get, and this pairing is occurring through hydrogen bond. So because of this formation of hydrogen bond between the two bases, one base is from the first polynucleotide, another base is from the second polynucleotide. So all these connection points, they are hydrogen bond. Once the hydrogen bond is formed, it will be now twisted. Okay, so the actual structure that is we know for DNA, that is for the second structure. Fine. So here we have the sugar backbone, which is this red strip, nitrogen base, you can easily see, and each turn, so if it is the starting point, it is basically the ending point. This turn is 3.4 nanometer. Fine. And here the groove that we are getting, two types of groove may be there. So this is minor groove and this is major groove. See, the distance is high. So major groove and minor groove, two types of grooves may be present, and this is the distance 0.34 nanometer, which is the distance between these two. Exact distance you don't have to remember, but try to have some little bit idea. Okay, that is, it is close, it is not very high, obviously it will not be very high, but it is very small. It is inside the nucleus, there is chromosome, then inside chromosome there is protein and nucleic acid. So you can understand how small it is. Fine. And the distance between these two polynucleotide, 2 nanometer. So these two strands are complementary to each other because the hydrogen bonds are formed not just between any bases. It forms between two specific pairs of bases. So as I have said, adenine always forms hydrogen bond with thymine, and cytosine always forms hydrogen bonds with guanine. And remember one more information, as there is presence of phosphate group, because this is the sugar phosphate backbone, pentose sugar that is associated with another that is each monomer unit, there is phosphate linkage. So this, because of this phosphate group, there is negative charge present. Remember, in the, if you look at this structure, there is always negative charge present, right? So because of this presence of negative charge, overall you can say that DNA is actually negatively charged. Okay, it is also true for RNA. Now we will see how these two bases, two combinations are possible. One is combination between guanine and cytosine, where total number of hydrogen bond 3. Remember, total number of hydrogen bond between guanine and cytosine 3. And when it is combination of adenine and thymine, that is AT. This is AT or TA. This is GC. So in GC there are 3, but here we have 2 hydrogen. And this is known as base pairing. This base pairing is actually responsible for the twisting of the structure. Okay. Here we have a question based on the structure of DNA. The reason for double helical structure of DNA. Remember, these two polynucleotides, they are not parallel to each other. They are forming some twisted structure. So it is because of which reason? 4 options are there. Electrostatic attraction, no, it is not true. There is no interaction between positive and negative charge. Van der Waals interaction, that is also not true. That is possible only when there are non-polar groups present. Then dipole-dipole interaction, that is also not true. Only correct option is the last one, that is hydrogen bond. Which base is present in RNA but not in DNA? Remember, there are total 5 bases. Under purine category, we have some bases. Under pyrimidine category, we have some bases. Under purine 2, under pyrimidine 3. Now, though there are total 5, but one base is not present in RNA, and that is uracil. And in one base is not present in RNA, which is thymine. And RNA is having only uracil, not thymine. But when it is DNA, it is the reverse case. So that is why if we ask which base is present only in RNA but not in DNA, it will be uracil. And if the question is reverse, let's say if it is asking which base is present in DNA but not in RNA, then it will be thymine. Now we will see some basic idea of RNA, though detailed structure of RNA is not required. Remember, it is single strand. And then we will compare DNA and RNA. So just like DNA, here also RNA, it will have some same type of structure. Only difference is now basic structure same, that is same 3 component nitrogen base, sugar phosphate unit. But difference is now we have only one strand. So it is single stranded. Often form complex and integrate base pairing interaction. So the base pairing that you have seen in case of DNA, here in this case it is not so simple, but we are not going into the detailed structure, that is not required, not included in syllabus. Then RNA molecules are 3 types. So just remember these 3 types, their exact detailed roles we are not going to discuss. So what are these 3 types? Depending on what type of function they are performing. They are mRNA because they are messenger RNA. Then ribosomal RNA, so they are called rRNA. And transfer RNA, they are called tRNA. Now if we see the difference between these 2, nucleic acid, it is double stranded, it is single stranded. It contains deoxyribose sugar, but in this case it is ribose sugar. Remember, in 2' position there is OH group present in case of RNA, but no OH, that is what is deoxyribose. And why we are calling it 2'? Because if you look at the base are also, there are some positions 1, 2, 3, 4. So again we cannot use 1, 2, 3, 4 for the pentose sugar. So that is why now we are using prime. Third point, it contains thymine as nitrogenous base, but in this case it is only uracil. It is genetic and hereditary material of the cells, but RNA is mainly involved in the synthesis of proteins. Roles are different. It is present in the nucleus of cell. Where it is present, that is also different for these 2. In RNA, it is present both in nucleus as well as cytoplasm. We know that when we consider any cell, there is nucleus, and then the rest of the part it is cytoplasm. So it is present both in nucleus and cytoplasm, RNA, but DNA is present only in the nucleus. So that is also important difference between DNA and RNA. So this part is finished. Now we will have some time, you can discuss some question based on the total chapter. RNA is different from DNA because RNA contains, we have 4 options. RNA contains, see, the first 2 options mention deoxyribose. Now deoxyribose, it is only for DNA. So these 2 you can easily cancel. Whether it contains thymine or uracil, that also you don't have to see. So deoxyribose, it is only for DNA. So RNA, it must be ribose. Now when it is ribose sugar, you have now 2 options, and among these 2 we know that thymine is present only in DNA, not in RNA. That means it is ribose sugar as well as uracil. So RNA contains cytosine and uracil as pyrimidine bases, but not thymine. But when it is DNA, cytosine is common, but now there is no uracil, only thymine. And guanine, adenine, that is common for both. So correct answer will be option 3. The number of oxygens, it is integer type of question, the second one. Number of oxygens present in a nucleotide. Nucleotide means it is monomer we are talking about, formed from a base that is present only in RNA. So it is asking the question indirectly. It is not directly saying which base is present. It is just saying the base which is not present in RNA. So in RNA, which base is not present? We know in RNA, thymine is not present. Okay. Sorry, which base is present? It will be uracil. So which base is present? That is present only in RNA. It is uracil. U. So considering uracil base, then ribose sugar or pentose sugar, and if you consider the phosphate backbone, considering all this, you have to check how many oxygens are present. So this is nucleotide. That is basic structure. We have 3 components: nitrogen base, it may be pyrimidine or purine, then pentose and phosphate group. Now it is saying that number of oxygen present in nucleotide formed from a base. Which base? Which is uracil, because that base is only present in RNA. So this is the structure of uracil, isn't it? Fine. So if this is the structure of uracil, now we have written the structure of uracil. This is the pentose sugar, and here we have this phosphate unit. Now if you count total number of oxygen, here we have 2 oxygen. In pentose sugar we have 1, 2, 3, total 3. And in phosphate unit we have CH2, sorry, it will be, there is structure is not correct. It is CH2O, then P double bond O, then O minus. So in the phosphate unit, total number of oxygen surrounding phosphorus, it is 4. So if we add all these, 4 plus 3 plus 2, then it will be total 9 oxygen atoms present. And if you are thinking that how to understand which pentose sugar we should take, because here we have taken ribose. So as we have, it is clearly mentioned which base, it is uracil. So when it is uracil, obviously you have to take ribose pentose sugar, because uracil is always associated with RNA. So you cannot take deoxyribose. So deoxyribose, if you take in this position, there will be H. But that is not correct. As you have taken uracil, so obviously it has to be ribose sugar. So that is why we will consider this H. So total number of oxygen 3 plus 2 5, 5 plus 4 9. The sugar produced after complete hydrolysis of DNA. We know DNA is deoxyribonucleic acid. And if you consider single monomer unit, that is nucleotide. In nucleotide, there are total 3 units. One is pentose sugar, then there is phosphate unit, and there is base. Now, when you are doing hydrolysis, these 3 components will be separated from each other. So you will get individual base, individual phosphoric acid. So that is why you are seeing this is always common. And the first part is sugar, the last part is base. Okay. Now it is DNA. That means in DNA, which base cannot be present? We know in DNA, this base cannot be present. So you can easily cancel this one. Now you are also one thing, as it is DNA, obviously the sugar unit will be deoxyribose. So these two also you can cancel. So now you are left with only one option, which is beta D2, it is actually 2 prime deoxyribose. Then you will get phosphoric acid, because now it is not phosphate, it is not connected, it is free. So it is phosphoric acid, and then H3PO4, and then added. So how we are cancelling? That is, here simply by cancelling some options you can reach the correct option. So as it is DNA, so obviously beta D ribose sugar not possible, 3 4 cancelled. Uracil not possible, option 1 cancelled. You are left only with option 2, which is the correct option. Next question. The number of carbonyl groups present in tripeptide. Now, tripeptide, which tripeptide? That is given ASP, aspartic acid, GLU, glutamic acid, and then lysine. So first we have to write the structure, because if you do not know the structure, obviously how many carbonyl groups are present, you will not be able to understand. And if you want to write the structure, we know that this is N-terminal. That is fine, because that is the general convention we follow. The first amino acid, this 3-letter expression that you are seeing, ASP, GLU, LOC. Left hand side is always N-terminal, this side is C-terminal. But you also have to know what are the side chains that are present in these 3 amino acids. So this is the structure of this tripeptide. This part, this is the side chain of aspartic acid. What is the side chain? CH2COOH. Then we have this peptide bond. Now the next amino acid is glutamic acid, where the side chain is CH2CH2CO2H. So this part is just like ASP, only difference 1 carbon extra. Then again peptide bond, total 2 peptide bond. This is first peptide bond, this is second peptide. And the last one, lysine, this is the side chain, CH2 whole 4, then NH2. So here we do not have any carbonyl, but in the first 2 side chain we have one carbonyl, and also in the C terminal, this is the C terminal, this is N terminal. In C terminal also you have 1 carbonyl, and in the 2 peptide obviously there will be 2 carbonyl. So total it is 5. The increasing order of pKa value of the following amino acids in aqueous solution. So here individual amino acid you have to consider when they are present in aqueous solution, their pKa. Now pKa is the dissociation constant of any acid in general if we say. So pKa is negative logarithm of Ka value. So if Ka is very high, that means acidity is very high. But when you take minus log K, there is negative sign, that means pK will decrease. So this is equal to pK. So when acidity is very high, then Ka is very high, but pK is reverse. Now it will be less. So lesser is the pK value, higher is the acidity. Remember this, it will help us to answer the question. Now if we want to know their Ka value, we have to know the structure. So these are the structures. For glycine, here we have hydrogen as side chain, and in aspartic acid we have side chain CH2COOH. That means it is acidic side chain. In case of third one, lysine, here we have basic side chain, and in case of arginine, again we have basic side chain. So obviously among these 4, this will be maximum acidic. That means its pK value will be minimum. So ASP, it should come at the extreme, that is least position, and that is present only in option 3. So this much information is enough. But why after ASP the next one is GLY? Because we have to arrange from low pK to high. So lowest pK will be for aspartic acid, because here the side chain is acidic. The next one is there is hydrogen side chain, that means it is neutral. So the next acidic will be this one, because these 2 are basic. Now among these 2, which one is more basic? Obviously arginine is more basic because of 3 nitrogen atoms. So lone pair availability is high. So here aspartic acid is acidic in nature, glycine is neutral, so it will come after this. And lysine and arginine both are basic, but arginine is more basic because of, as you can see from the group. So considering all this, correct option should be this one. Fine. Enzyme related question. Match list 1 with list 2. We have total 4 enzymes. Now enzyme, in most of the cases you will see, if you look at the name, you will find some similarity with the substrate of the corresponding reaction, because each enzyme is responsible for some particular reaction. And in that particular reaction, if you know the substrate, then you can find some similarity with the name of the enzyme. Not just always substrate, sometimes we will see whatever process is going on, or that is depending on that, you will find some similarity with the enzyme's name. So this will help you to answer the question. So look at the name of the enzyme and also look at the substrate. Maltose, substrate, it is converted to glucose. So for this conversion, the enzyme, the matching name is this one. So D must be associated with 2. And if D is associated with 2, that you can find only in option C. So see, this much of information is enough to answer the correct option. Choose the correct option. Okay. So this is that is maltose into glucose conversion. Now, if it is correct, then you can easily consider the other list 1, list 2 combination. So for invertase, that is the first one, is cane sugar, which is nothing but sucrose converted to glucose and fructose. And here the name of the enzyme is invertase because inversion occurs. That is, from plus sign we get plus and minus sign. So that is why there is inversion going on. So that is why the name of the enzyme is invertase. So here it is not based on name of substrate, but based on the process, whatever is going on. Then zymase, glucose is converted to ethanol and CO2. So invertase, it is matching with 4. A is related to 4, B is related to 3. Glucose into ethanol. Diastage, that is starch to maltose. So don't think that here product is maltose, so it is matching with D. No, that is not the case. It is matching with substrate. So substrate is maltose in option, number 2. So that is why D should be associated with. So option C, that is the correct option. Okay. One question, we'll quickly discuss it. We don't have much time. When sugar X is boiled with dilute H2SO4 in alcoholic solution, 2 isomers we are getting from X. A on oxidation with HNO3, the first isomer, it can be oxidized by HNO3, you are getting sulfuric acid, and the second isomer is levorotatory in nature. We have to find what is the structure of X. Number 1, maltose, if you do hydrolysis, if you get 2 moles of glucose, that means both A and B same. So this cannot be correct option because we are getting to individual isomer, individual molecule, different molecule. Sucrose, when you are doing hydrolysis, you get glucose and fructose. So this can be correct option. What about other option? Last one, it is not at all correct because starch is polysaccharide. You cannot get after hydrolysis 2 isomer. You will get more than that. Lactose on hydrolysis yield 1 mole of glucose, 1 mole of galactose. Okay. Now when it is sucrose, this is sucrose, it is undergoing hydrolysis. So these are the values for optical rotation. From plus, now it is plus for glucose, negative for fructose. If you want to know the details, that you can also find in the respective part of the that is our discussion. You can find the details, and among these only glucose, which is A, that is undergoing oxidation, and B is levorotatory, which is clear from the sign. So this should be the correct option. But why not lactose? Maltose starch already we have considered, but why not other one? Because when it is lactose, if it is matching with A, that is fine. Glucose is giving you saccharic acid, but the second one is plus sign, so it is not levorotatory. So that is why this is also not the correct option. Sucrose is the correct option. So we are at the end of the session. Thank you for listening.